Good morning. I'm James Daubenspeck. Moonlighting proteins, and really that's about the only time I'm going to use that word because it sounds rather flippant, and this is a way larger and way more important topic than that. Multifunction proteins are really ubiquitous, guys. They're everywhere in every life form that you can think of, from synth all the way up to us. Now, there's a number of ways this can happen, right? And I'm not going to spend a lot of time on this because I don't have a lot of time. So we first found this phenomenon in 2016 in Mycoplasma pulmonis. This is published from there. This molecule right here attaches to the protein and links it to the membrane surface. Okay. This is immunopurified enolase from Mycoplasma pulmonis in this SDS page right here. This is from the membrane. This is from the cytoplasm. You can see that the Kamasi stains, glycostains, phosphostains, and then that's knocked out in the cytoplasm. Now, if you plotted this on a structural model, that particular modification from Mycoplasma pulmonis, it's right at the dimerization domain. This is what it looks like in the cytoplasm. It's an octamer. It's the penultimate step in glycolysis. But then when it's found on the membrane, at least in pulmonis, one of the linkages is right here. Now, if you plot what we found in Syn3.0, there's this one, there's another one here, there's one on the back side up here, and there's one on the other side, which really kind of made me rethink that particular theory. So if we look at enolase from Syn3.0, and this is just a gas chromatogram with the MS on the back end of it. This peak A is mannose, and remember this is membrane purified enolase. This is the mannose phosphate, and I can tell these from the mass spec signatures. These are very characteristic of these molecules. And then this particular molecule is the lipid, and this is from Syn3.0. Okay. Now we know some of these enzymes are on the outside of the cell, and this is just antibodies that were raised against either Staph aureus enolase or human ENO1 and reacted with whole cells from JCVI-Syn3.0. You can see we get a very strong reaction with Staph, and although it's kind of mild, we get a reaction with the ENO1 to the ENO1 antibodies. Now this is the molecule that we have found. In the parent GM12, this is a rhamnophospholipid. Rhamnose synthesis is knocked out in Synt, and so it becomes, this molecule just doesn't finish the final step of synthesis and is a mannophospholipid, and that's what we find in Syn3.0. Now, we've also looked at databases from Bacillus cereus and some from the gram-positive cocci. And we have found evidence for this molecule in those databases as well. These are publicly available mass spec databases. So we think this molecule is responsible for multifunction proteins, not only in Synt, but in mycoplasmas and molecules. And I would go as far to say it's out all the way through most of the firmicutes, so almost all the gram positives. Now, if I'm right, and this is that common a molecule, how in the world did the mass spec world miss this for 40 years? The answer is fairly simple, actually. On most protein setups, when they're doing mass spec work on peptides, without fail, they have an HPLC on the front end to separate out the molecules to make it a little easier on the mass spec. Now, this works real good because it separates, but really based on hydrophobicity of the peptide. However, almost all of them have a reverse phase column that they use. Well, this part of this molecule basically welds itself to the reverse phase part of the mass spec column. And what ends up happening is this molecule shears. You can imagine if this part of it is stuck to the column and it's under pressure from the flow of the HPLC, this molecule will break right here, and we end up with this molecule stuck to the peptide as the modification. About 1 out of 5 times we'll see it as a disaccharide. And how do we know this is occurring? Well, we know that the molecule on there is a rhamnophospholipid from Page's work and from the GCMS. And the HPLC on the front end of the mass spec is an HPLC, right? So you can plot that just like you would a regular chromatogram and look at it on the screen, and that's what's up now. If you plot a regular peptide without a modification, you get a standard looking chromatogram peak. If you plot a peak with this modification on it, you get this mess from the stochastic nature of this molecule shearing off the reverse phase column. Okay? So what do we need for this pathway to be active? All right? We got a modification of the protein with a rhamnose or a mannose in the case of synth. I think that the enzyme in mycoplasma, now this was probably not true in all the firmicutes, but I think in mycoplasma, it can link, one enzyme can do all 5 amino acids that I've got on board: threonine, serine, tyrosine, asparagine, and glutamine. Right? There's a pattern that I've seen in our data, but it's really complicated. There's a consensus sequence, we think, but the amount of variation in the sequence controls the percentage of a protein that gets modified and sent to the surface. You need something to transport that modified protein now to the inner leaf of the membrane. You need enzymatic linkage of the modified protein to the phospholipid that's held in the membrane. And then you have to somehow transport that to the other side. And we know that a lot of these proteins are on the outside of the membrane from antibody work. So at the moment, we have identified 30 spectra in JCVI-Syn3.0 that I am comfortable publishing, and we are in the process of doing so. We have 100 more spectra that I believe are real, but for one reason or another, either I can't tell whether there's a difference between a threonine and a serine that are in the same sequence, or there's a tyrosine there that I can't tell the difference with, or we've lost part of the fragmentation pattern of the Ys or something. There's some reason why we can't publish them. Now, that's 26% of the genome, right? And now I will admit that at least in mycoplasma's genitalium, the proteins like the adhesins are held on with this molecule as well. So some of these may not be multifunction proteins. Currently, proteins with 1 or 2 copies on the surface, really low population proteins, we're probably not picking up. The mass spec proteomics data that we're using for this work, and my colleague John Sandberg is going to talk about it after I do, is already published. It's in the PRIDE repository. There's the identifiers. This is my shameless plug. We just had a paper come out Monday, and it's on surface hexosylation in Syn3.0. Like I said before, the ratio of monosaccharide to disaccharide mannose phospholipids seem to be about 5:1. Now, whether this is an enzymatic mistake or is wired into the system, maybe the doubles are on the inside of the membrane, the singles are on the outside, something like that. My kitty cat. We have identified 4 disparate amino acids in the enolase sequence that are modified with the mannophospholipid in Syn3.0. There are other known multifunction proteins that have multiple linkage sites. These represent a distinct fold and another function. Now, if I've got a few minutes left, let me check. I got a few.
You got a little time, James. Go ahead, go for it.
let's just join me in the thought experiment for a minute. Okay. So we've got our protein sequence, right? I don't believe this thing is folded and then transported across the membrane. I think that with some of these larger proteins, that's going to cause major issues. So it's either a linear sequence being escorted by a chaperone, now linked to the inner leaf of the membrane, right? Or it's partially folded, like certain intrinsic alpha helices within the sequence fold without even looking, right? But then if you've got multiple linkage sites, what happens to it once it hits the outer membrane? The outer layer gets attached to the outer layer of the membrane and now has to refold, okay? So if it's linked here or here or here, each one of those linkages is going to lead to a different fold, right? And we have found 4 of them in the Syn3.0 enolase. So what this could theoretically mean is that enolase has at least 6 functions that I can think of, if that's even close to true. That could be a different situation where you have 2 linkages, right? Or 3 or 4. Maybe enolase has a specific shape that it wants to form into. You can imagine how 2 linkages would change the fold of that protein, right? And we have multiple examples of this. Enolase is that way, EFTU is that way, where we find multiple sites for this modification on these proteins. Like I said, this data set is published. Y'all can go look at it if you've got mass spec software and are curious. I would love to collaborate with some sequence structure prediction people on this idea. Like I said, I've got the linkages that we have for Syn, so they will be published. We're going to try to get it out by the end of the year. It's part of John's, my colleague who's coming after me, dissertation, so it's definitely coming out. In conclusion, I'd like to thank everybody from UAB and everybody at the JCVI. It's really a pleasure working with you guys, and this bug just keeps getting more fascinating and fascinating. So with that, I'll be happy to take any questions if we have time.
James, you've got a couple of minutes. So one thing that comes to mind for your proteins that appear to be on the surface of the cell. So once they leave the cell, once they're on the outside, they're in oxidative conditions. And so if there are 2 cysteines in a cell, in a protein, you're going to get another kind of fold.
True. Very true.
But if no one online says anything, I think that we have colleagues at MIT that I identified when we were working on that proposal that I failed to get in last year that we can work with, and I know Liam would be interested in doing this. And we can think about removing some of the, maybe changing sequences of some of these proteins and see if making them so they're not going to go into the membrane kills the cell.
Right.
Because there could be
doing some cryo-EM experiments on 3A and tagging the enolase with an iron labeled antibody, and then doing cryo-EM on the different enolases on the surface and see if there is any difference in the fold in that molecule on the surface of 3A. That's something we've discussed, and certainly we have the capability of doing it.
James Pelletier's got a question.
Yes,
Amazing work, James. Just a brief question of clarification. So the proteins you detected by this method are exposed outside the cell, or could they also be associated with the inner leaflet? They're just the ones that are exposed, no?
We have data on 3 or 4 that are conclusive that they're on the outside.
Okay, thank you.
Those are mainly some of the more standard multifunction proteins. So enolase, DNAK, EFTU. We have antibody proof that those are on the outside of the cell.
Okay.
I would assume that some of these are on the inside, right? Because we're doing just really a membrane purification. So it's high-speed centrifugation to separate out the membranes, basically. So we can't really tell the difference.
Yeah, hi. I have a quick question. First, I apologize. I lost connection in my middle of my answer to James, so I'll call you James. But a quick question for you is, based on your work, can you estimate how many of these degradasomes you might have? Because we're using as a model for the degradasome, when we have it in the minimal cell, it has an enolase, and then it has the RNase Y, and then two exonucleases or actually three, that can read and chew up the mRNA from either end. So how many of the, we have an excess of enolase, so I think the excess still doesn't quite count for all the degradasomes we build.
Right. From what I can tell, and this is just a feeling based on mass spec work, about 80% of it goes into the glycolytic cycle. About, I would say 5% goes into the degradasomes, and about 15% is on the surface, would be my estimation.
Interesting. Wow. Okay, it's a new feature. We don't have this model.
Enolase is on the surface of every cell. I mean, every cell. Even the ENO1 from humans is on the surface of our cells.
So, some of those essential proteins, we don't know their function. We do know from Hamilton Smith's work that they are membrane proteins. So it could be this is one of the things being exported.
Absolutely.
Very
Folks,
cool. If we could validate that, boy, would that kudos.